Good morning everyone. In today's video, we will gonna learn about the pure cultures of bacteria. Now, what is the pure culture? Pure culture is basically bacterial colonies comes from a single colony. Okay. This is the concept of pure culture. Now, suppose we want to isolate pure cultures from soil. So first we will collect the soil sample. Okay, dilute it. Then spread it on petri plates containing bacterial growth media. Incubate simply at 37 degrees centigrade for 24 hours in BOD. Okay, now after 24 hours we got a result. Now what is this result? How the bacterial plate looks like after the incubation? Suppose this is a petri plate. Okay, so there are different types of colonies will appear from the soil sample, because soil is basically a very good source for the microbes. So we get different types of colonies, like few are looks like this, some are like this, and some types like this. Okay, so there are numerous colonies, and different types of colonies can be grown on a media when we isolate the bacteria from the soil sample. Now, if we want to get pure culture from these colonies, then what we have to do? Again, we have to make the growth media, pour it into a petri plate, a glass petri plate or a plastic petri plate, whatever it is. Then, with the help of an inoculation loop, we will suppose we will streak this green color colony. So we incinerate the loop well with the help of a burner and 70% ethanol inside the laminar airflow, and then we just streak it on the bacterial growth media. How we will streak? Suppose this is a plate from the top view. Okay, this is a plate. Now we want to isolate only green colonies. So we will, after incineration, the loop, this is a loop. With the help of this loop, we just pick these colonies and we will streak it on the bacterial growth media. How? We first streak it like this. Okay, then again incinerate with the help of the burner, cool it down. Again, from the edge of this, we will again streak it like this. Okay, again incinerate, cool it down, and then after incineration, the final streaking is like this. This is known as streak plate method. Okay, this streak plate method, specifically this is a four-way streaking method, is very, very important to isolate pure colonies of bacteria. Means the colonies or a group of colonies that will appear from a single colony like this. Now in case of blue, we have to pick it from the blue colony with the help of this loop, and again we have to streak it on another growth media containing a different plate, not in this plate, different plate. In this case we again have to do it like this. And in case of this black color colony, we have to do this, then this, and finally this one. So in this case, we will get 3 different types of pure culture. But in reality, there is not only 3 types of pure culture, there are numerous cultures are present. Means various types of bacteria are present in a growth media. If we want to isolate the bacterial cultures from the soil. So this is a streak plate method, which is a very important method, and the pure culture technique is basically used or developed by a very eminent German bacteriologist who is known as Robert Koch. And due to this streak plate technique method, many types of pathogens, bacterial pathogens, which are responsible for human bacterial disease can also be isolated as a pure culture. Okay, so this technique is very, very useful, and it is also used in the microbiology labs to isolate the pure colonies after the initial isolation of the bacteria, whether from the soil or from water sample or from air, whatever. So this is a very efficient technique basically. Hope you like this video. Thank you so much.